Hello and welcome to SAM online class. This is Dr. Subrat Kumar Mohanty. Friends, today we shall discuss about prokaryotic genome organization. One of the distinguished characteristics of prokaryotes is the absence of extensive internal compartments. Extensive internal compartments means various membrane-bound organelles like nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi complex. These are the membrane-bound organelles. In prokaryotes, these membrane-bound organelles are absent. Another important characteristic of prokaryote is that they are monoploid. All prokaryotes are monoploid. That means they have a single copy of the genome. We are diploid. That means we have two copies of the genome. So prokaryotes are monoploid. They have a single set of genes. Prokaryotes can be distinguished into two very different groups on the basis of genetic and biochemical features. These two very different groups are the bacteria and the archaea. Most of the commonly encountered prokaryotes are the bacteria. The bacteria include different groups like gram positives, gram negatives, cyanobacteria and many more. Second group is the archaea. The archaea is a group of prokaryotes that are less studied and have mostly been found in extreme environment. The genome of prokaryotes are very different from eukaryotic genome. If we compare the genome of largest prokaryote and smallest eukaryote, there may be some overlap in size. On the whole, the prokaryotic genomes are much smaller. Take an example. In E. coli K12, the genome is just 4639 kilobase and the small genome has only 4405 genes. In addition to this, the physical organization of the genome is also different in prokaryotes and eukaryotes. The traditional view about prokaryotic genome organization is that an entire prokaryotic genome is contained in a single circular DNA molecule. But this view is not applicable in all prokaryotes. Prokaryotes have additional genes on plasmids. Plasmids are the extra chromosomal DNA molecules that are capable of replicating independently. Some of the plasmids can also integrate into the bacterial genome. This plasmid is known as episomes. So the plasmids which are present in many bacteria provide some added advantage to the microorganisms. Plasmids are not essential for bacteria. So plasmids are dispensable. Dispensable means without plasmids, bacteria or prokaryotes can exist effectively without them. In prokaryotes like E. coli, the genome is unipartite. Other prokaryotes are more complex. Take an example of Borrelia burgdorferi B31. In the prokaryote Borrelia burgdorferi B31, the genome is multipartite. In this prokaryote, there is a linear genome which contains 911 kilobase and this linear genome carries 853 genes. In addition to this linear genome, there are additional 17 or 18 linear or circular DNA molecules. These 17 or 18 linear or circular molecules together contribute another 530 kilobase and 430 genes. So the genome organization in the microorganism, that is Borrelia burgdorferi, is different. In Borrelia burgdorferi B31, the genome is segmented. Not only in Borrelia burgdorferi, in many other bacteria and archaea, the genomes are multipartite. Friends, we are discussing about the prokaryotic genome organization. In prokaryotes, there are more genes and less space. Some genes have virtually no space between them. Take an example. In E. coli, 3 genes. One is THRA, another is THRB, and the third gene is THRC. These 3 genes together constitute the threonine operon that is concerned with the synthesis of amino acid threonine. The gene THRA is separated from THRB by a single nucleotide. Between these 2 genes, there is a single nucleotide. Whereas between THRB and THRC, there is no space at all. And prokaryotic genes are shorter than the eukaryotic counterparts. The average length of a bacterial gene is about 2/3 of the eukaryotic gene even after the removal of introns. Similarly, bacterial genes are comparatively larger than the archaeal ones. In prokaryotes, for example, in E. coli, introns are absent, whereas in eukaryotic genes, introns are present. What is introns? Introns are the non-coding sequences that separate the coding sequences. E. coli has no discontinuous gene at all. What is discontinuous gene? The gene which contains both coding sequence and non-coding sequence is known as discontinuous gene. In E. coli, there is no discontinuous gene at all. Another important characteristic of prokaryotic genome is the infrequency of repeat sequence. That means repeat sequences are not frequent in the prokaryotic genome. Most prokaryotic genome do not have high copy number genome-wide repeat families, but such type of repeat sequences are seen in very large number in eukaryotic genomes. But the prokaryotic genomes contain certain sequences that might be repeated elsewhere. Example of these certain sequences are insertion sequence and transposable elements. Insertion sequences are present in the genome of E. coli. In the genome of E. coli, 2 insertion sequences are seen. One is IS1 and the other is IS186. In prokaryotic genomes, transposable elements are also present. Transposable elements have the ability to move around the genome. Most prokaryotic genomes are less than 5 megabase in size. Regarding prokaryotic genome organization, the traditional view is that an entire genome is contained within a single circular DNA molecule. This traditional view is applicable to E. coli and other commonly studied bacteria, but it is not applicable in some cases. The circumference of E. coli chromosome is 1.6 mm, whereas The size of the E. coli cell is 1.0 into 2.0 micrometer. This indicates that E. coli chromosome, which is a single circular double-stranded DNA molecule, is squeezed into a tiny space within the E. coli cell with the help of specific DNA binding proteins. Specific DNA binding proteins help in the packaging of the E. coli genome so that the genome will be fit into a very small space within the E. coli cell. Most of the information related to the organization of DNA in the nucleoid comes from the study of E. coli. In E. coli, the genome is supercoiled. There are two types of supercoiling, positive supercoiling and negative supercoiling. Positive supercoiling occurs when additional turns are introduced into the DNA double helix. Negative supercoiling can develop when turns are removed from the DNA double helix. The torsional stress that is introduced by overwinding or underwinding in a linear DNA molecule is easily released by the rotation of the ends of DNA molecule. By rotation of the ends of a linear DNA molecule, the torsional stress is easily removed. But this is not possible in a circular DNA molecule because a circular DNA molecule has no free ends. In this situation, the circular DNA molecule responds by coiling around itself to form a more compact structure. For the bacterial genome, supercoiling is an ideal way so that a circular DNA molecule can be packaged into a very small space within the E. coli cell. So the supercoiling that is seen in the bacterial genome is generated and controlled by two different enzymes. These two different enzymes are DNA gyrase and DNA topoisomerase. DNA gyrase and DNA topoisomerase are the two different enzymes that are responsible for generating and controlling supercoiling in the circular DNA molecule. The E. coli DNA molecule or the E. coli genome, it do not have unlimited freedom to rotate once a break is introduced. After introduction of a break in the circular DNA molecule of E. coli, the DNA molecule do not have unlimited freedom to rotate. The current model regarding the structure and organization of bacterial genome is like this. According to the current model, in the structure of bacterial genome, there is a protein core. This is the protein core. From this protein core, you can see about 40 to 50 supercoiled loops. These are the supercoiled loops. This is the protein core. From the protein core, about 40 to 50 supercoiled loop radiate out. These are the supercoiled loop. The supercoiled loop have radiated out from the protein core. Particularly this loop, it has been broken. So in this loop, there is no supercoiling. In this loop, the supercoiling has been released by introducing a break. So by introducing a break, the supercoiling can be removed only in this loop, not in all the loops. Each loop contains approximately 100 kilobase of supercoiled DNA. In each loop, the DNA is present in supercoiled form, and each loop contains 100 kilobase of supercoiled DNA. When we introduce a break in one loop, then the supercoiled DNA that is present in this loop only, that will be opened up. Next important point is, like eukaryotic genome, in prokaryotes also, the genome is packaged with the help of specific DNA binding protein. The protein component of nucleoid. What are the protein components of the nucleoid? The protein component of the nucleoid include DNA gyrase and DNA topoisomerase. DNA gyrase and DNA topoisomerase are the two enzymes that generate and control supercoiling. In addition to these two proteins, there are additional at least 4 proteins which play a specific role in packaging the bacterial DNA, because bacterial cells are very small in size. So the bacterial genome has to be packaged into a very small space. For this packaging, specific DNA binding proteins are necessary. So for packaging of the E. coli genome, at least 4 different proteins believed to play a specific role in packaging of the bacterial DNA. Out of these 4 different proteins, the most abundant protein is Hu protein. Hu protein. The Hu protein acts in a similar way like that of histone protein. Histone protein helps in folding of the eukaryotic genome. Hu is the most abundant protein present in the bacterial nucleoid, and this Hu protein, it forms a tetramer. The Hu proteins form a tetramer around which approximately 60 base pair of DNA becomes wound. So friends, this is about the organization of prokaryotic genome. For more updates, please subscribe SAM online class. Thank you very much. See you again. Bye bye.